Today we will work to describe for you the functional components of a muscle contraction. In order to understand the activities that will take place, we must first outline some of the key players. The muscle, made up of fascicles, which are little bundles of muscle fibers, surrounded by a paramecium, a layer of connective tissue. Made up of muscle fibers are myofibers, which are made up of the most simple element, the myofibril, the contractile part of a striated muscle fiber. And the sarcomere being the basic functioning unit of a myofibril. And finally, mitochondria, responsible for the conversion of food to usable energy, which allows for muscle contraction. To begin, an electrical signal called the action potential, originating from the central nervous system, is transmitted via a dendrite past the cell body to the axon. This signal must pass through the fascicle, which is a bundle of muscle fibers, all the way down to the myofibril to achieve its final goal, muscle contraction. The action potential propagates by activating sodium channels, depolarizing the membrane as the impulse moves along the axon toward the knob. This movement allows calcium to flow into the axon via the calcium channels. The calcium influx causes synaptic vesicles containing the neurotransmitter acetylcholine to migrate toward the plasma membrane. This migration releases acetylcholine out into the extracellular space between the motor neuron terminal and the motor end plate of the muscle fiber. Acetylcholine will bind to its receptor on the motor end plate of the muscle cell. The binding process will open the sodium-potassium channels, or pumps, and via these anti-port pump operations, sodium will move into the cell, and potassium will move out. Because the channel is more permeable to sodium, the muscle fiber membrane becomes more positively charged, triggering an action potential. The action potential travels along the sarcolemma and down the T-tubules. From the T-tubules, the impulse passes to the sarcoplasmic reticulum. The action potential spreads through the muscle fiber's network of T-tubules, which continues the process of depolarizing the inner portion of the muscle fiber. As the impulse travels along the sarcoplasmic reticulum, the calcium gates in the membrane of the sarcoplasmic reticulum open. Calsequestrin is the main calcium-binding protein inside the sarcoplasmic reticulum and acts as an internal calcium store in muscle. The release of calcium bound to calsequestrin through calcium channels trigger muscle contraction. Calcium fills the binding sites, represented in red, in the troponin molecules, represented in black. This alters the shape and position of the troponin, which in turn causes movement of the attached tropomyosin molecule, represented in blue. Movement of tropomyosin unblocks the binding sites and subsequently permits the myosin head, represented here in green, to contact actin. Contact with actin causes the myosin head to swivel. During the swivel, the myosin head is firmly attached to actin, so when the head swivels, it pulls the actin, and therefore the entire thin myofilament forward. This swiveling action repeats as long as ATP is available as a result of the mitochondria and calcium is present on thin filament. And as they do, the thin myofilament is being pulled by the myosin heads of the thick myofilament. Thus, the thick and thin myofilaments are actually sliding past each other. As this occurs, the distance between the Z discs of the sarcomere decreases. As sarcomeres get shorter, the myofibril, of course, gets shorter. And obviously, the muscle fibers and entire muscle get shorter. At the end of the swivel, ATP fits into the binding site on the crossbridge, and this breaks the bond between the crossbridge, myosin, and actin. In each sarcomere, thin myofilaments extend in from each end. Thick myofilaments are found in the middle of the sarcomere and do not extend to the ends. Because of this arrangement, when skeletal muscle is viewed with a microscope, the ends of a sarcomere, where only thin myofilaments are found, appear lighter than the central section, which is where the dark, thick myofilaments are found. This is why skeletal muscle is called striated muscle. The alternating light and dark areas look like stripes or striations. The light areas are called the I bands, and the darker areas, the A bands. Near the center of each I band is a thin dark line called the Z disc. The Z disc is where adjacent sarcomeres come together and the thin myofilaments of adjacent sarcomeres overlap slightly. Thus, a sarcomere can be defined as the area between Z lines. The central portion of each thick filament is connected to its neighbors by proteins of the M line. These dark staining proteins help stabilize the positions of the thick filaments. In a resting sarcomere, the H zone or H band is a lighter region on either side of the M line. The H band contains thick filaments but no thin filaments.